of the bacterial DNA. And the other thing about the bacterial DNA is huh?
We were trying before that.
No, that's no, we can start. Yeah. We did. Where you work? Where you work?
We haven't done it. We didn't have side that you were on. We had like 3 more points at the bottom on that.
Which one? On this? Ah, shoot, sorry. On this one?
Yeah, we finished it.
We did not finish this one, no?
Right. No, we talked about DNA replication is a very accurate process.
Okay, so DNA replication is a very accurate process. Yeah, I was going to catch up on that part, you know. So DNA replication is semi-conservative. We talked about the semi-conservative over here. This is a semi-conservative DNA replication, where every time a DNA divides, the daughter cells receive at least one of the original strands. Then we also talked about the fact that the DNA replication is asymmetric. That means one of the strand is always synthesized at a faster pace compared to the other strand. And that's why we have the leading strand versus the lagging strand of the DNA synthesis. We are going to talk about the leading strand and the lagging strand today in a little bit more detail, as well as the components of the DNA replication replisomes. DNA replication is a very accurate, DNA replication is bidirectional. We talked about it last time. And lastly, the DNA replication is a very accurate process because of the proofreading activity of the DNA polymerase. The DNA polymerase has a proofreading activity. So there's some editing activity. Every time it puts up a new nucleotide base, so if it is putting up a new nucleotide base against this A and T, G and C, T and A, and every time it puts up a complementary base, it makes sure that it is a correct base that is put in. If there's a mistake, it steps back, it removes the mismatched base, and then puts the correct one and then moves on. And that's why we say replication is a very accurate process because of due to the proofreading activity. Due to the proofreading activity of the DNA polymerase. So DNA polymerase, it maintains a high fidelity of replication. Any misincorporated bases are immediately removed, and it is the new correct base is incorporated in the DNA replication. Otherwise, just imagine what happens if the DNA replication it doesn't have a proofreading activity, if it is not an accurate process. Just imagine your skin cells which are being shedded and they are being replaced. Just imagine it like your nose cells will be growing on your skin. How creepy is that? Okay? Or some other cells will be growing on your face. That doesn't happen because the DNA replication is a very accurate process, and that accuracy is maintained because of the action of the DNA polymerase, which has a high fidelity and a good proofreading activity. DNA replication also requires energy. It's not a free ride. The DNA replication requires energy. And who provides the energy for DNA replication? Huh? Who provides the energy for the DNA replication? Nope. Oh, don't trust what I write here. Who provides the energy?
Isn't that just the cell itself?
The cell has to do it. I was expecting somebody will say ATP.
Oh, I said that.
You did?
But I said it really low because I'm not confident right now.
Oh no, I want you to be confident.
Thanks to that quiz.
But guess what?
Oh, no.
ATP.
That's why I'm not confident.
That's why it was whispering. Hello.
The first part is, you may think that ATP provides the energy for the DNA replication, but the truth is, ATP doesn't provide the energy. Every nucleotide that comes in to get incorporated, it comes as a DNTP. So it comes as a DNTP. So it comes in as CTP, deoxy-CTP, deoxy-GTP, deoxy-TTP, or deoxy-ATP. So whichever nucleotide needs to be incorporated, it brings its own energy. And that's why we tell it as NTP. That's why we have it here, DNTP, which is a generic name for the deoxynucleotide triphosphate. So every DNA, every nucleotide which is to be incorporated, it must bring its own energy. And that energy, which is being brought in by the nucleotide, it is always coming as like similar to the ATP. So when the CTP comes in or the GTP comes in, there are two of the high energy phosphate bonds are present. So here in this, it is the DGTP. So there are two of the high energy phosphate bonds are present. When these are made free, it provides it energy. And that drives the G to form a phosphodiester bond with the 3' or with the 3' OH. So every time the DNA, which needs to be replicated and an incoming nucleotide comes in, every time it comes in, it comes as a nucleotide triphosphate, but then only this part. So this part is the only one with one of the phosphate group gets incorporated and it forms a phosphodiester bond. Sure. So every time a nucleotide comes in, it comes as a triphosphate. The last two phosphate group, because they are high energy phosphate like this or shown over here, this is the high energy phosphate bond. So these two bonds are broken and that provides it energy to form this phosphodiester bond between the 5th carbon atom and the 3rd OH group of the deoxyribose sugar. So this bonding is called as a phosphodiester bonding, and that's how the DNA chain elongation occurs.
Is that where the 5' and 3'?
That's where the 5' is always the blocked one. The new nucleotide always goes in the 3', 5', or 5', 3' because there's a OH group over here. At the 3', there's a carbon, there's an OH group, and the new incoming nucleotide forms the phosphodiester bond there. The next part is replication initiates. It does not initiate randomly, but there are specific areas on the DNA from where the replication initiates. And that is called as the origin of replication. So DNA the replication process always starts not randomly, but from a defined area, which is called as the origin of replication. So DNA replication is not a random, the initiation is not a random thing, but it starts from a specific area, which is the origin of replication. And the replication process by itself is a collaborative effort of many components. Some of them are proteins, some of them are non-proteins, but they're all complex together as what we call it as replisome. Complex as replisome. The next slide talks about the replisome. What are those? Which is there in the next slide, and we are going to talk about that in a second. Now, the other thing about the bacterial DNA is that one of the things which you need to know is the bacterial DNA is methylated. There's a methyl group that is added mainly to the C and the A groups, mainly to the A as well as rarely to the C. So A is mostly, but C is rarely. They are methylated. That means they contain a methyl group in addition to all the other functional groups that are present within the DNA. And this methylation of the bacterial DNA has some significance because that methylated DNA portion is involved in the control of gene expression. Significance is, and it's in your textbook on page number 205. In some cases, some of the genes which are methylated can be turned on. Some of the genes which are methylated can be turned off. So bacterial gene expression. So that can basically control or regulate gene expression. It could be either turning on the gene or turning off the gene, depending upon whether they are methylated, the genes are methylated, or they are not methylated. They contain either the methylated adenine or the methylated C residues. At the same time, the methylation of DNA also is the starting point. At the origin of replication, where the initiation begins, those areas are heavily methylated. So initiation of replication is also from those methylated DNA from a methylated DNA area. The third significance of the methylation is it allows the bacteria to degrade the viral DNA but do not disrupt the bacterial DNA. So it protects the bacterial DNA, but it would degrade the viral DNA. Anytime a virus goes inside, the bacteria might try to degrade the viral DNA because the viral DNAs are non-methylated compared to the bacterial DNA. So these are some of the significance of the methylation of the bacterial DNA.
Can you repeat the first one?
Yeah.
I know the second one.
Is the regulation of gene expression. Some genes can be turned on following methylation. Some genes are turned off following methylation.
The viral DNA is not.
And the viral DNA is non-methylated, so they are preferentially degraded. But the bacterial DNA, which is methylated, is protected. Degraded by the bacterial nucleases.
You said a gene can be turned on or off after, like at the methylation
Yeah.
point, that's when it.
Not after the methylation point, like some of the genes that contain the methylated sequence, those sometimes they are used to turn on the gene, and some of the genes can be turned off because of the methylated sequence. So regulation of the bacterial gene expression is mainly regulated by the methylation process. Now the next slide talks about the replisome, I believe. And we'll talk a little bit about the replisome. And in this context, we'll also talk about one of the antibiotics that is often used for controlling bacterial infection by targeting the DNA replication process. So replication, as I mentioned earlier, includes both protein as well as the non-protein components. The non-protein component includes the DNA template. So one strand of the DNA which needs to be replicated, that's a non-protein component. Then the RNA nucleotides. because the RNA nucleotides are used as a primer, and I'm going to talk about those things in the next picture, folks. What are the primers? And I'm going to catch it up because this is a primer of an RNA sequence. Energy, because energy is required for the DNA replication process. It's an energy requiring, what do you call it, as an anabolic reaction. It requires a deoxynucleoside triphosphate because those are the nucleotides that are incorporated in the DNA molecule. It also requires the participation of the origin of replication as well as the primers. So these are some of the non-protein components of the DNA replication process. What is the role of the primers and all? We are going to discuss this part, so don't get panicky, and I will explain it to you to the best of my ability so that you are clear on that part. So the protein components, these are the non-protein components which are here where I just mentioned it. There are some protein component of the replisome that facilitates the replication of the DNA. And the replisome protein contains could be either with some enzyme activity associated with the protein, or they could be non-enzymatic. That has no enzyme activity, or there's no inbuilt enzyme activity associated with the few proteins. Those non-enzymatic protein includes the single-stranded DNA binding protein, which is also referred to as a stabilizing protein. It ensures that once the two strands of the DNA are separated, they don't snap back. As in this picture, if you look in this picture, these are the stabilizing protein. These are the stabilizing protein that binds to the separated strands of the DNA. You see that? So these are the stabilizing protein or the single-stranded DNA binding protein. They prevent the hydrogen bonding between the separated strands. Then you also have the DNA polymerase. And I'm going to talk about DNA polymerase in a lot more detail in the next slide, where you have about what is the role of the DNA polymerase. So just bear with me for a second. Then you have the helicase. The role of the helicase is it helps to separate, it helps to break the hydrogen bonds and separate the two strands. So the role of the helicase is breaks the hydrogen bonds between the strands and separates them. So the bond between the two, the hydrogen bond between the two strands, those are broken by the helicase, and it separates the two strands. In this picture, the next picture over here, in this picture, the helicase is the one that is breaking the hydrogen bond between the two strands. So now the two strands get separated from each other. You also have the action of the gyrase. What the gyrase does, can I erase this part, folks? Did everybody write it? Ari? Did you write it? The gyrase, what it does, it alleviates the tension built up between the separated strand. It alleviates the tension buildup between the separated strands, and it does that by nicking one of the strands.
By what? By making?
by nicking.
Oh.
By nicking one strand. I think I did explain this thing to you a few days back in the lab. Do you remember with the rubber band?
Oh, yeah.
Do you remember? So that is the role of the DNA gyrase. And there are certain antibiotics which belongs to the ciprofloxacillin group. It targets the DNA gyrase. So antibiotic like ciprofloxacillin, it targets the DNA gyrase. So ciprofloxacillin targets the DNA gyrase of the bacterial cells. Cipro is often used for the treatment specifically for the UTI, for the urinary tract infections. Cipro is one of those favored antibiotic, but it is never given to pregnant women because, so this is given for UTI treatment. And you know about the anthrax. Did I talk about the anthrax that day? That when we had that anthrax problem in Washington sometimes back, anthrax can be easily treated with penicillin antibiotics. It's a gram-positive bacteria. You can treat it with penicillin. But those individuals were treated with Cipro just because you don't want to take a chance and figure out later that they were using penicillin-resistant strains. So what they did was they treated it with ciprofloxacillin. Those individuals were treated with ciprofloxacillin. So it can be used for the treatment of anthrax, just in case you don't want to start treatment and then realize, oh my God, those were penicillin-resistant strains of bacteria. You need to be a little bit cautious in giving ciprofloxacillin to pregnant men. Oh shit, what am I doing? Pregnant women. Oh, it's not my day today. McConkey to McMonkey, pregnant men's, woman to men's. Sure. So, it's never given to pregnant. No, women. It is not given to pregnant women because there are some serious side effects. Because of some serious side effects, it really impacts the cartilage development of the newborn or the unborn child. That's one of the reasons why ciprofloxacillin is never given to pregnant women, because it targets it, or it basically it has effects on cartilage development. So I'm trying to catch up on this one, so you know what is ciprofloxacillin and what are some of the side effects, and it basically targets the DNA gyrase. Is that clear, folks?
So for nicking one strand, what does that mean to nick? How do I understand?
Oh. Does anybody have the hairband or hair? Can I borrow?
Yeah.
Can I borrow a hairband from someone, folks? Okay. Alrighty, folks, here it is. So the DNA, if you recall it from last time, is very supercoiled, and during the DNA replication, the two strands need to be separated like this. And the separation of the two strands is mediated by the DNA helicase. But there's always a tendency that the two strands will again come back together. Okay? Now, how would you prevent this from happening?
Cutting it, so it cuts.
So you cut it, and that cutting is done by the DNA gyrase. It cuts one of the strand. That's what is the nicking of
Okay.
the strand.
Oh, got it.
Yeah. Now, the primase is the one that makes the RNA primer. So this one makes the RNA primer, makes the RNA primer. Then you have the ligase, which joins the Okazaki fragments. Ligase is the one that joins the Okazaki fragments. Lastly, you have the RNase H. The RNase H, I'll write it here. The RNase H is called as the RNase H because it only degrades the RNA portion of the RNA DNA hybrid. So RNase H, the H is for the hybrid of DNA and RNA, and it degrades the RNA portion. That's the RNase H role. Before I go into the complete the play of the DNA replication process as is shown in this picture, again, this picture looks a little bit scary, but I will try to make it as easy as possible, okay? So don't be scared out on this one. Before I get on to this one, I just want to catch up a little bit on the DNA polymerase, because the DNA polymerase is like the quarterback of the replication system. The DNA polymerase is one of the most important player of the DNA replication. And the DNA polymerase has certain properties. There are some specific properties associated with the DNA polymerase as such. It uses always one strand of the DNA to make the other strand. That's one thing which is universal. It always uses one strand as the template to make the complementary second strand of the DNA. It has a proofreading activity. That means there is some editing functions which are inbuilt within the DNA polymerase. It's something like this is a template of the DNA, and I'm the DNA polymerase, and I will incorporate, so look at this. This is the primer, because I cannot start from here. I cannot start from this point. There is an RNA primer here, which is complementary RNA sequence. I'm the DNA polymerase. What I do is I will put up the next complementary strand, and I will keep on extending the primer by adding complementary nucleotides. So I'll keep on extending the primer. And if I find, oops, I made a mistake last time. Instead of a C, I put a G here. So what it does, it backtracks, and that's where the editing role of the DNA polymerase is. It backtracks, it removes the misincorporated or mismatched nucleotide, replaces it with the correct nucleotide, and then it moves on. And that is what is the proofreading exonuclease activity.
So it's a dimer?
Huh?
A dimer that does it?
Not a dimer, so this is the DNA strand. I'm putting up the new nucleotides here, and the new nucleotide, and I realize, oops, I made a mistake, and I made some boo-boo in the last one. So takes a step back, removes that nucleotide, adds the correct nucleotides, and then again moves forward. You see that? So that's what is the proofreading. It's called as an exonuclease because it cannot go all the way down here and then realize, oh my God, I made a mistake there. It doesn't do that. It just goes at the end. That's why it is called as an exonuclease versus the endonuclease, where it can go anywhere in between. It elongates the DNA only in one direction, and that is always from the 5' to 3'. 5' to 3' direction, and that is the prime reason we have asymmetry in the DNA replication process. So it always extends the DNA in one direction, in the 5' to 3', and because of this activity, we have this asymmetry with the leading and the lagging strand in the DNA replication process. One strand is synthesized at a faster pace compared to the other strand, which is made at a slower rate. Not to mention, it can only add nucleotide. The DNA polymerase only adds nucleotide to an existing fragment of nucleic acid. And this existing fragment of nucleic acid is nothing but it's an RNA primer. So think about it, I'm the DNA polymerase, I keep on extending, this is the RNA primer, I keep on extending the RNA primer by adding complementary nucleotide to the DNA template. So that's my job, that's the job of the DNA polymerase. But at the end, this primer needs to be removed because this is the RNA primer, and who removes that is the RNase H. Because this is a DNA template, this is the RNA primer. So RNase H will remove this RNA primer, and the DNA polymerase, once of the DNA polymerase, will fill it up with the DNA nucleotides, and the DNA ligase will now connect the two. This whole sequence is shown in the previous slide. But before I get on to that one, I just want to talk about the replication fork. This replication fork is the part where the active DNA replication is occurring. So once the DNA separates, this is where the new nucleotides are being added. This is the lagging strand. Then there will be a leading strand which will be chasing the fork, and that's where it's called as replication fork. And in the replication fork, obviously, the parental strands are no more paired. They are no more paired. And remember, in a replication bubble, which is shown here, in a replication, this whole thing is a replication bubble. In a replication bubble, there is one replication fork. They're going in two separate directions, but then they will come together. So this whole thing is a replication bubble. If you took it that way, this whole thing is called as a replication bubble. And in a replication bubble, there are two replication forks in the opposite direction. They go in the opposite direction, and there are two replication forks in the replication bubble where active DNA replication occurs. So with all of these, with this background, I think it's time for some little break. So when you come back from the break, we'll talk about this. We'll talk a little bit about the transcription and gene expression, and then we'll talk about the genetic code, and we'll move on to that.
What about under the chart for polymerase 3 and 1? The ribosome?
Yeah, when I'm going to talk about the replisome, it will catch up because the DNA polymerase 3 is the one, and the DNA polymerase 1 is when it replaces those nucleotides in the primer. That is done by the DNA polymerase 1.
Like next year before.
Huh? This one?
Like, no.
Oh, this is an entertaining one. You'll like it, this one.
Our brains are not functioning at full capacity right now.
It's not functioning? Okay, I'll make it some lighter thing. But we are not going to do the whole thing, like just a little bit and then I'll stop.
Dr. B, what can I do other than doing the best I can on my homework assignment as labs to ensure that I can get a GPA score? I feel like I walked in the lab exam feeling really good about it, and then
But didn't you do good?
I got a 72%.
What do you mean that's not out of 100?
So, no, I got
Whatever number you have is out of
out
75.
of 75. Yeah, I got a 72%. So like,
Really?
yeah, and I
don't
really thought
believe that,
I really
no.
thought I had done better than that. So I
I
felt
made a
pretty
mistake,
good
but I
about it.
thought you did good on
I
that
thought I
one.
did good too. And now I
Talk about
let me
exam
go.
3.
no, the exam we just took.
That's why.
Yeah. I'm like
I just
so
knew I did so
worried.
wonderful on that, and I did not.
I
I walked out like, oh, I had that, and my test scores did
and I'm
not
like,
result.
it's hard because I want to go to office hours, but I work like 60 hours a week.
Oh,
So I'm doing the best I can with studying and doing all the homework and everything, but I'm just wondering if there's anything else I could do because I'm supposed to apply for nursing schools this summer, and I just it's not your problem, and also I'm like, help me.
You know what I would suggest is if you can take the last week off from work, tell them you will work extra hours. You
Yeah,
will work on the weekend to substitute that one.
I don't know if I can do that.
Let me first of all check your.
Yeah, and
did I
like-
put it wrong? It's possible.
Yeah, I don't know, and I like
make
if
a
you
mistake
can
in-
even help me calculate, so I know how well I need to do on
Sure.
both the lab
I'll
exam
do
as well as the final. Because I mean I'm gonna study as much as I can in the next 2 weeks, but I'm just at this point like I just want to do well, and I've
No,
been
no,
trying
no,
so
no.
hard.
It's not even 2
Sure,
weeks,
no,
it's a
I'll
week.
do one
Ah,
thing.
it's a week.
It's a week.
Oh my god.
That's what I'm saying. Yeah, the final is next
Yeah.
week. Yeah. I need to calculate everything, and I missed the last quiz. That was the day I found out
You
about my
should be
niece.
able to retake. You should be able to take that. You
Quiz?
heard her story.
Quiz.
It's
I
not
dropped
retakeable.
one of the quizzes.
I know, so-
But you had a genuine reason I can give you the quiz.
Okay. When can I come and do it? Like tomorrow? Like when?
Tomorrow will be the best part.
What time?
I'll tell you.
Okay.
Yeah. Okay. Let me do one. Give me a moment, folks.
Okay. And then, like even this week, I was just kind of in my feelings, so I didn't study like I should have. Because all of my family got together, and like I can't run to Florida.
Right, of course not.
So anyway, it is what it is. I mean, this test though that we just took, I was like, I got this. I'm like,
Really?
I know this is my strongest one,
Really?
and it was.
You were strong
When we were studying, that's what I said.
Just in lab too. I was like, you're doing the number out of 75, right?
Yes,
Not 100.
that's what.
Okay.
I looked at what he gave, what I got, and I'm like, well, wait a minute.
I wonder if there was a
knew
mistake
this.
made because I feel like you probably should have done better than I did.
That's what I knew. Like,
You
I
did.
knew
You
this.
did.
That's what I'm trying
You
to
knew
figure
this
out.
better than all of us did.
I'm trying to figure out how the hell I got the score
What
I
score
got.
did you get?
59.
Percent? Or 59 out of 70?
59 out of 75. I don't understand how I got a 59 out of 75.
I think there was a mistake.
I've
on
got
the
to
scan
see it
process.
because there's got to be a problem.
No,
Because I'm like,
because
I
did
knew
better than
this.
you. You knew way more than I did.
I knew this. So that's why I'm like, what the hell? So that's like a 78. That's a 78 point
So
something. There's
much
no
better
way
than
I
that.
got a
There's no
78 on that last
Yeah,
exam. I'm like,
I just want to look at it side by side.
I've got to look at that. I've got to look at it because I'm like, there is no freaking way.
That
I
doesn't make
knew
sense.
that material.
Yeah, you did.
If I didn't know anything else, I knew I knew that material.
Yeah, technical problems happen all the time. I wouldn't be surprised.
I'm just like, I don't,
Because I saw
don't get
it.
it. And that's kind of like what I was like, wait a minute, when we were studying, I knew the answers
More
to
than all of us.
everything.
Yeah,
How
you
the
did.
hell did I I think maybe I wasn't getting more it didn't really enough, but I know what I really that was I could at least see it look like 60. Like I saw that but it's not. I mean, still, I mean,
No, I
So if it would be helpful, you're welcome to. That's like 25. Yeah, I know. Haven't had these. Anyway, let me know.
Also, I definitely went into that knowing about it.
Oh, wait.
I don't know, it's just so good. I just like it.
You just need to get at least an A or B.
Like,
Yeah, A or B. No, I don't have, I don't
and I have
I'm pulling you.
Oh, no. You're not gonna be
I'm
able
literally
to.
gonna be out of the weekend.
All the planes are like sitting back.
It's Sunday, I'm not even starting I mean, I can stay Monday. I'll be back.
Otherwise, I'm
What did you Oh, you're not certified. I'm not certified. Like you have them. Yes. I said for. Are you certified? You're not certified.
But I think we have a propane one now. I think I'm gonna get it. But he wants just our
I'll give it to you.
Okay. Dr. B, right, what's the class I was
Huh?
the class I was like,
Total points?
Yes.
Check the syllabus, is there.
no, I mean as of right now, before the
Alrighty, folks, let's start. So, class, let's do this part. Let's do this part because I need to synchronize, and next class period I'll be talking about HIV and a few things about the antibiotics, the several acting antibiotics and the sulfur drugs and some of the ideal properties of antibiotics. So I would like to finish as much as possible, and if I'm not able to do it, we'll stop it there, okay? So let me start this one. So this is for the DNA replication. And all of these three pictures, because here it shows the initiation of the DNA replication. This picture shows the synthesis of the leading strand, and this part shows the synthesis of the lagging strand of the DNA. So initially, because the DNA replication process has these three steps, remember the DNA replication has the unwinding, the complementary base pairing, and lastly is the bond formation. Those are the three steps of the DNA replication process. Now, when you do the unwinding, that is mainly done by the DNA helicase. So initial process, these two get separated. The stabilizing protein binds to this one. It also prevents the reformation of the helix is prevented by the stabilizing protein to a certain extent. Now, then come the leading strand and the lagging strand. The leading strand is a continuous or an uninterrupted polymerization of the DNA nucleotides, and it is always trying to chase the fold. So it starts again from here. It has to first make up an RNA primer, which is the two of those things. What I put it over here, that's the RNA primer. And the DNA polymerase keeps on extending the RNA primer as is shown over here. There is a 3' OH group over here, and the new nucleotides which are coming as a nucleotide triphosphate, and the two phosphate groups are released. It provides that energy so that not the hydrogen bond, then it can connect to this OH group over here, free OH group. So every time the leading strand is a continuous polymerization, and it is trying to chase this fold. It is always trying to chase this fold. It is moving in the 5' to 3' direction, and it starts from the origin of replication where you have this RNA primer, and it keeps on chasing the fold. It's an uninterrupted polymerization of the DNA nucleotides. That is the leading strand. The lagging strand is the one which is made on the opposite side, and it is a discontinuous DNA replication. It's a discontinuous because if it starts from here, and if it tries to do this way, then what happens is, by the time the fork comes here, this has started from here, then what will happen to the other part of the DNA? So that was a kind of a big time question for quite some time. And Raji Okazaki, in his honor, we call this as Okazaki fragment, from MIT, he first saw, using a very high magnification microscope, he saw the DNA replication and he found that there are two. One strand is being synthesized at a faster rate, which is the leading strand, and the second strand is synthesized in small pieces, and that's what he called it as the lagging strand of the DNA. And that is the time we also explain how the 5' 3' action of the DNA polymerase, how can it be satisfied in explaining the DNA replication process. He just passed away, just a few years back he passed away, and till the time he passed away, he was still a member of the MIT group of scientists, and in his honor, we call this as Okazaki fragment. Look at this, what happens. When it ends up, let me pull it down this one. Look. What happens is, this DNA, it opens up. This part is synthesized by the DNA polymerase, so this is the leading strand, it's not a problem. Are you all okay with this leading strand? Now for the lagging strand, it starts from here with the RNA primer. So it provides a 3' OH and the DNA polymerase 3. That DNA polymerase 3 will be providing the nucleotide and it will keep on extending up until this part. That's why the lagging strand is slower replication, because it is made in small pieces. Piece by piece by piece it is made. And here also look at this, RNA primer was made and that was extended all the way up until here. And then this will be joined, once the RNA primer is released, then it will be joined to this segment by the DNA ligase. Okay, and again, when this comes all the way down here, this primer will be removed, and that will be filled in by the DNA polymerase, will replace the RNA primer with the DNA nucleotides, and then these two fragments will be joined by the DNA ligase. And because this is done in small segments, it is a slower process, and that's why we call it as the lagging strand of DNA synthesis. So there's a leading strand and there's a lagging strand. The lagging strand is the one that is done at a slower pace. It's a discontinuous replication. And Okazaki, there are many Okazaki. Here you will see there are a lot of Okazaki fragments are being made. And all of these Okazaki fragments, like this is one Okazaki, this is second Okazaki, then they are all connected to each other by the DNA ligase. I don't want to tell you anything more on this because I don't want to put up too much of pressure. I understand completely, folks, there are other things to be talking about. So I'll stop about the DNA replication at this step.
You said they're in between the DNA ligase, but it looks like they're in between the
What is that?
It looks like they're actually in between the RNA primer.
So here's what happened. The RNA primer has to be removed by the RNase H.
Right.
Because in a DNA, you cannot have an RNA sitting in the DNA.
Gotcha.
So what happened? The RNA primer will be removed, and that gap, these 3 nucleotides need to be filled by the DNA polymerase 1.
So the RNA primer is just a fill-in.
RNA primer needs to be filled in, and that is taken care of by the DNA polymerase 1.
Got you.
And DNA polymerase, like this is the template. If I'm the DNA polymerase, I cannot start from here. I just cannot start from here.
Everyone just danced. I just thought it was okay, just in case there's
I'm just thinking, I don't care.
Don't do it with these girls. That's the first thing
You made me scared.
Should we go now? I was like,
When it on it up there.
That's scary.
Someone like abandoned their bed. Was like right by your house.
They're all trying to figure out how much it would. And I was like, it went
It was not the big one. It was the ghost.
They said it was over highlighter. I feel like ours.
These are someone. Is that, are they your?
I just heard your voice and spoke to remember, like, I was so little and I could tell you, like, my outfit ever since then, I can't look like a kid. I go to, like, different levels. Oh my god, so
Okay, Paul, here's the deal. Yep, I'm scared too. I'm scared too, so we'll do one thing. We'll just wrap it up this one.
oh my gosh. People running out.
So class, let me quickly do this slide and then we can leave.
Oh, okay.
So the next one is the gene expression. I know, just take a deep breath. No, there will be a lab because we need to complete something. So I will be very short on this one.
I think that was a sign that that's a wrap.
Huh?
I can't even concentrate.
I know, folks. I'll be very short on this one. You don't have to know everything. You have to know everything. The gene expression. I know. No, my heart is also like beating fast. Hands are shaking. So the gene expression includes two of the processes. One is the transcription.
Wait. We don't even know that. It's not going to
I know, like, I just like that.
It includes the transcription and the translation process. And these are two separate processes, but they're interdependent in the sense translation cannot occur until transcription is done, And once the transcription is done, translation is inevitable. So these are two independent but separated processes. And during the process of transcription, an mRNA is synthesized. During this process, mRNA is made. You don't have to know these ones, this one you don't have to know. An mRNA is made in the process of transcription, and the DNA acts as a template. So from the DNA, the RNA is made, and that is the process of transcription. And translation is when that mRNA is to be coded into the protein sequence, a protein amino acid sequence. It is to be the amino acid sequence of proteins. So that is the translation, and this part is shown over here in a much better way. So this is the DNA. From DNA, then it's not that the DNA is converted, but the DNA, one strand of the DNA is copied. That is the most important word. DNA is copied to the RNA sequence in the transcription process. And once it is transcribed, once the RNA is made, invariably it will be translated by the ribosome to the amino acid sequence of proteins. You have to go back because this transcription process is done by the RNA polymerase. And RNA polymerase is different than the DNA polymerase in the sense the RNA polymerase has very poor proofreading activity. It has a very poor proofreading activity, and at the same time, the RNA polymerase, which makes the RNA from the DNA template, it also makes it in the 5' to 3' direction. And in the RNA, there is, in the RNA per se, the one which is the product of transcription, the RNA is always single-stranded. It's not a double-stranded RNA. It is always single-stranded. It is not a double-stranded. That's one thing. There are 4 types of the nucleotides. There are 4 types of the RNA are being made. Those 4 types includes the messenger RNA, the transfer RNA, the ribosomal RNA, and the primer RNA. The mRNA, tRNA, rRNA, and the primer RNA, and these 3 are involved in the translation.
What's rRNA again?
Ribosomal RNA.
And T?
Transfer RNA. So ribosomal RNA, transfer RNA, and the messenger RNA, they are involved in some or the other way in protein synthesis. Whereas this one, the primer RNA, is involved in the DNA replication. This is involved in the DNA replication. You still have the purines and pyrimidines, but there is no T. There is no thymine residue in the RNA. And RNA you have just the ribose sugars. There's no deoxyribose. In the RNA, you have the ribose sugars, but not the deoxyribose. Okay? I will stop it here, folks, for today. We'll see next week, next Wednesday morning. We are still in the shop. And I realized one thing today, folks. In this big classroom, if something happens, we cannot get out of it. This is something I really realized today. That will help you understand. Otherwise, you have to memorize it.
If I just memorize that, well,
No, it will not help because my question is no. Yeah, no, we can do the lab now.
Question about the exam on Wednesday. Is it going to be like the stations? Is it going to have questions?
Yes, I'm going to talk.